Hey, Bio30s Manchak here. I am recording this video with a slightly different setup again because I wasn't overly happy with how things turned out on the C4 video. So this is your C5 Part 1 video, and I'll just show you right away here exactly what that's going to look like. So we are doing C5 Part 1 right now, which is this bit right here. I'm recording it on my mic here just because I want to look goofy. No, because I think the sound's going to come out a little bit better for you. So this is the first part of C5. Like I said before, it's a long concept. It's going to take us a while to get through that concept. So I wanted to break it up into two pieces to give something a little bit more manageable for you guys to work on. This is going to have us working through basically our use of Punnett squares, which allows us to predict and determine probabilities for whether an offspring will turn out one way or another based on the parents that are being bred together. And then secondly, it's going to run us through something called a pedigree and get you guys understanding that. The textbook pages, again, are located at the top of your notes for this particular section for Concept C5, and there'll be a couple of videos I'll make reference to for this particular section, and that is these two here. Again, the Amoeba Sisters do an amazing job of explaining this stuff, often far better than I do, and so I want you guys to follow that. So, without further ado, I'm going to jump into our concept that we're working on after I move this out of the way, and we'll play from this slide. So what you guys should have done already now is you should have already watched concept C4, which was on our introduction to Mendelian genetics and how all of that works. And you should have an idea of what the terms gene and allele mean. You should have an idea of what dominant versus recessive means, homozygous, heterozygous, genotype, phenotype. What we're going to do now is we're going to take what Mendel taught us and we're going to start to try to put it together into something practical for us to work with so that we can get a sense of how genetics works and how things are passed down from one generation to the next. So that's what we're dealing with here, is we're going to try to describe the evidence that was investigated by Mendel, and then we're going to start to do some comparing of ratios and probabilities of genotypes and phenotypes for, in this case, dominant and recessive traits. And then we're going to get into the more complicated stuff with multiple and completely dominant, codominant alleles later on. So the probability inheritance of single traits. Mendel's work on pea plants allowed him to begin to predict what offspring would look like if he knew the genotype of the parents. So if he knew that this parent had these alleles and this parent had these alleles, he would with relative confidence be able to predict what the offspring would look like if he took those individuals and bred them. He couldn't tell you exactly that if we took, one gamete from each of these individuals, fertilized it, he couldn't tell you exactly what the organism would turn out like, but he would be able to tell you that if you had many offspring from these two parents, he could give you a good idea of the probability of having this kind of individual, this kind of individual, this kind of individual. So he could predict some probabilities. So his predictions were based around these probabilities and knowing the genotypes of the parent plants. So what he was able to determine were phenotypic and genotypic ratios. And these ratios, these terms should sound reasonable to you, right? A phenotype is the physical expression. So a phenotypic ratio then is the ratio of offspring with the dominant trait to the recessive or alternative trait. So in other words, would we have 3 round-seeded individuals to 1 wrinkled-seeded individual, or would it be, one, or would all the offspring be round-seeded, or all the offspring be wrinkled-seeded? Okay, that would be determined there. The genotypic ratio would be the ratio of offspring with each possible allele combination from a particular cross. So what would be the ratio of homozygous dominant to homozygous recessive to heterozygous? Or big R to little r to big R little r. Does that make sense? Something along those lines. So those are our ratios that we're dealing with. The probability of an outcome of a particular trait is easily determined using this formula. This is not complicated math in any way, shape, or form. P stands for probability. And so probability is the number of ways that a given outcome can occur divided by the total possible outcomes. And right now we're going to start really simple. We're going to have 4 possible outcomes in the Punnett square that we use. And then you just have to decide, how many ways did our given outcome occur over the total outcome. So it's going to be something over 4 that we're dealing with to start with. So speaking of Punnett squares, we can use a simple tool called a Punnett square to help us predict the phenotypic and genotypic probabilities of various crosses. In other words, we can use a Punnett square as a tool to determine what are the possibilities of individual A being this, and individual B being this, and individual C being this, based on the parents that we have up above. So, if you haven't seen one of these before, this is a Punnett square. And a Punnett square, like I said, is a simple tool used to represent alleles from the parents. So I'm going to just take my pen out here and we'll draw all over the place a little bit. So a Punnett square for the simple alleles or simple crosses that we're doing right now will simply just be a 4 section square that were created. And what's happening is what we have to know are the genotypes of the parents. And so I attached the little tail here to say that this parent is male and this parent is female because these look a little bit like sperm. And when we do this cross, then we remember the law of segregation, which means that the father and the mother's alleles will separate, will segregate during meiosis 1, and we will end up dividing these two apart from each other. So when using a Punnett square, the alleles from each parent are separated, as they are here. So we're sort of thinking that there's a dividing line here and there, and that this indicates the segregation that occurs in meiosis 1 and helps us to predict possible trait outcomes for each offspring. So this is a cross of a homozygous dominant round pea plant, so that's the R for round pea, versus the recessive wrinkled pea. Something I didn't say in the last concept that I'll mention now is we often use, we choose the letters based on what the dominant trait would appear like. So since our dominant trait is round, we would choose the letters R, big R and little r to represent our alleles for this particular gene, which is the shape of the pea. So that's why we have big R, big R. So homozygous dominant, that's this individual here, and homozygous recessive, that's this individual here. And using a Punnett square, what we can do is we can predict what the offspring are going to turn out like. So if this gamete here came together with this gamete here, that would produce an individual who is big R, little r. I hope that makes sense. Okay, and if this gamete here came together with this gamete here, that would also produce an individual that is big R, little r. And as you probably predicted, if this gamete here came together with that gamete here, we would have another big R, little r situation and a big R, little r situation here. So I want to circle back for a second and I want to talk about the terms we discussed on the last slide, which were phenotypic and genotypic ratio. Well, there ain't much of a ratio going on here. Okay, for our genotypic ratio, we would say that these individuals are 100%, right? For our genotype, 100% the offspring would be 100% big R, little r. And our phenotypic ratio would be that they are 100%. Okay, there's no real ratio here. These individuals are going to be 100% round. Okay, I hope that makes sense and where that came from. Okay, so those are the ratios. So let's get a little more complicated as we move through here. So let's jump to the next slide. I'll do an example. A breeder crosses a pea plant with wrinkled seeds and a pea plant with round seeds. She knows that the plant with round seeds is heterozygous for the gene for seed shape. The allele for round seeds is dominant over the allele for wrinkled seeds. And so we need to figure out the expected genotypic and phenotypic ratios. Pardon me, throat's dry today. I think it's allergy season. So I'm going to get started on this with you guys. So let's select my pen and let's kind of go from here. So what do we know? We know that they're crossing a wrinkle-seeded plant with a pea plant with round seeds, but they specified for us that that round-seeded plant is heterozygous. So that means, and I'm going to set up my cross here, that means that we have a round-seeded plant that is heterozygous being crossed with a wrinkle-seeded plant. And since wrinkled seeds are recessive, then that individual has to have a genotype of little r, little r. Okay, so I hope that makes sense there. So then what we're going to do is we're going to set up a Punnett square to do this cross. Now you don't have to draw the egg and the sperm like I did on the last one. All you need to do is just make sure you separate out the gametes from each other. Okay, so this individual here is up here. This individual here is over here. Then I just create my 4 sort of separate cells here, and we can go from there. This comes together with that to make a big R and a little r. This comes together with that to make a little r and a little r. Big R and little r come together. Okay, and 2 more little r's. Okay, hope that makes sense. One thing I should note, and I haven't mentioned yet, is if you have a heterozygous individual, like these two are here and this one was here, always list the dominant allele first and then put the recessive allele second in that heterozygous individual. So the question really, so we've done our Punnett square. Here's our proof of what the cross is going to look like. So when we bring these two individuals together, these are the offspring that we could accept if they had only, or expect. This allows us to predict probabilities. I have to be clear here, okay, is if we bring these individuals together, we could have 50 individuals that were big R, little r that were produced. But out of 100, we should expect pretty darn close to 50 individuals being little r, little r. So really all this is doing is predicting probabilities for us. So what are our genotypic ratios and our phenotypic ratios? So I'm going to do G for genotypic. Okay, our genotypic ratio then is going to be, right, this is, remember we figured out our probabilities, right? Our ratio is going to be 2/4 for big R, and 2/4 for little r, little r. Another way we can write this, and I think it's better to write it this way, is that we have 2 individuals that are big R, little r, and 2 individuals that are little r, little r, which we can reduce down to 1 to 1. Okay, so for every 1 individual that's big R, little r, we'll have 1 individual that's little r, little r. Okay, that's our genotypic ratio, and I always like to add underneath just so that we understand that that's what this is that this is the ratio that we've created. 1 big R, little r for every 1 little r, little r. Okay, and then what is our phenotypic ratio here? Well, this one's relatively similar, right? We're going to have, remember, big R produces round individuals, little r, only little r present produces wrinkled individuals. So we would again have a 2:2 ratio of round to wrinkled. Okay, but it would be better if we put this and reduce the ratio down 1:1 round to wrinkled. Okay, So I hope that it makes sense. The key pieces of doing one of these questions for me that I want you to do: write down the original cross. Let's do this in a different color so it shows out a little bit better. Write down the original cross, okay? Construct your Punnett square, and then do your ratios. All right, let's jump to the next slide. I want to talk about test crosses, and every once in a while this would pop up on the diploma exam, and really all it is a specific term. A test cross is the cross, and this is very specifically what it is: cross of an individual that has an unknown genotype, okay, to an individual that is fully recessive, okay? And the reason why we can do that is that we can figure out using this test cross what the unknown genotype is based on the probabilities that we know, right? Because there's three possibilities right now at this point in the unit that the unknown genotype could be. So our first cross, our first possibility is it could be big R, big R. Our second possibility is it could be big R, little r. And our third possibility is it could be little r. And if all of those are being crossed with little r, we can do a Punnett square for each of these individual crosses, and we would get very different phenotypic and genotypic ratios. Okay, what this would do then is allow us to figure out this unknown genotype. For example, if we did this cross here without constructing the Punnett square, I would know that I would get 100% of whatever the dominant trait is. Okay, 100% physical expression of the dominant trait. If we did this cross here of the big R with little r, we would get a 50-50 ratio of the dominant to recessive expression. And then if we did this cross here, the two homozygous recessive, we would get 100% of them being recessive. Okay, I hope that makes sense. That's why a test cross is so powerful, is that you could take an individual you know is fully recessive and then take that unknown individual, breed them a bunch of times and see what comes out. And if you breed them enough times and you see only dominant is coming out, you know that unknown individual is this. If you breed them enough times and you see it's roughly 50-50, you know that that individual was this. And if they come out 100% recessive, you know that individual was that. I hope that makes sense, and the power of something called a test cross. Here's another example of something we can do. A horticulture worker has seeds from a particular cross, but has no information about the genotype and phenotype of the parents. So he grows the offspring and records the trait of each offspring in the table below. What is the genotype and phenotype of the parent plants? So what we can do is basically reverse engineer a Punnett square. And I would do this if I was tackled or given this question as a possibility to tackle. I would do this sort of as a guess and test. I would look at these numbers first. And looking at these numbers, I would say that 5,472 is about, and it's not perfect, but it's about 3 times the 1,850. So I would say that we would have about a 3-to-1 ratio of, in this case, round to wrinkled. So if we have a 3-to-1 ratio, what possibilities of parents is going to produce a 3-to-1 ratio? Well, one possibility is one parent could be big R crossed with little r. Okay, and if we did a Punnett square, and I'm not going to do every Punnett square to save us some time, but if we did this Punnett square in a really messy fashion, I think it should be obvious to you that all of the individuals here are coming out to be round. Okay, so they're all going to come out to be round. So it can't be that Punnett square. Okay, it can't be that cross right here. If we tried to cross an individual that is heterozygous with an individual that is homozygous, and we did the Punnett square, you would find that this would be about a 50-50 ratio. But we don't want 50-50. We don't want 1-to-1. We want 3-to-1 round to wrinkled. So that's not going to work. So as we continue our guessing and testing, we may happen across a situation where both parents are heterozygous. So big R, little r, big R, little r for both parents. Okay, and what we're going to produce here is this sort of Punnett square. I'm sorry this is getting messy. I'm trying to rush a little bit here. Okay, and writing with a mouse is never a good thing. If you look, 1, 2, 3 of these individuals turn out to be round for every one individual who turns out to be wrinkled. So I would suggest then, based on the ratios that we've established here and the numbers that we were given here, that the parents were likely both heterozygous for this trait. Hope that makes sense, how you reverse engineer that a little bit. So at this point might be a good opportunity for you, if you'd like to, if you're done listening to me for a bit, hit pause and have a look at page 604 of your textbook, or better yet, jump into the worksheet Punnett squares and probabilities. And I'm just going to call that up right now for you and show you what that worksheet looks like, so you can get a sense to what it entails. So this is the worksheet here, and it's really going to move you through a lot of the stuff that we've covered already, from deciding whether this genotype is heterozygous HE or homozygous HO. If purple flowers are dominant to white flowers, what are the phenotypes going to be? Or brown dominant to blue, those kinds of things. Then you'll get slowly into constructing Punnett squares, and you're looking for percentages or phenotypic and genotypic ratios in here. And then you'll do some practicing with that. So I would recommend, if you've got the time, to go do that right now and do a little bit of practice, hit pause, and then come back to the rest of this. And by the way, there's a key up on the Google Classroom as well, that you'll be able to see all of the answers that I wrote in there. So hit pause now if you want to do that. In the meantime, when you're ready, I will be ready to move on to pedigrees, which is the next concept. So hopefully you've gone through and done some of the practice work, and now we're going to just quickly run through talking about pedigree charts and what pedigree charts do. And basically, I'm going to look at it this way. A pedigree chart is about the same sort of level as something like a Punnett square. It's a tool. It helps people who study heredity and genetics to understand how things are passed from one generation to the next. And in particular, a pedigree chart is really valuable for us to work our way through a whole family tree of individuals and sort of sort out where traits move through within that family of individuals. So a pedigree chart is another tool in the arsenal of genetic analysts. Okay, and it's used to record the transmission of a particular trait or traits over several generations. Usually we're dealing with just one trait because it's simpler that way. And I'd like you guys to kind of think of a pedigree as a very simple family tree. Instead of just saying, hey, this person married this person and that person was this person's parent and blah, 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 we're going just a little bit more detailed, and we're talking about a particular trait, a physical expression of something. So how are they passed from parents to offspring, and then from offspring's offspring to new offspring, and so on. And the chart shows all of these using a series of symbols and shaded patterns. And so one thing I will say right now is this comes straight out of what would be a data booklet for Biology 30 that you would get on an exam. And this is something that as you guys are working online right now, I will post up into the Google Classroom so that you guys can have access to this while you're going through your quiz. And it's located. Let's see if I can pull it up. I'll show you where it is um, on that document that I was talking about. So there's a data booklet that I'll post up, and it's got a bunch of information on it, formulas and things that we'll get to later on. But here it is on the second page, and it's just a legend of what we have going on for pedigrees. So I'm going to jump back to the presentation and we'll look at it in there. So pedigree charts are based on logic and require a little practice to use. So sometimes this gives people a little bit of trouble because you have to employ a little bit of logic to understand what's going on, and you have to have a decent understanding of um, you have to have a decent understanding of how simple monohybrid crosses work. I needed to pop my collar for some reason. I don't know. That's uncomfortable. So you need to have a simple understanding, a basic understanding of how a monohybrid cross works, the stuff that we've just been working on. So here's a guide to what we're going on. If you find a square box on a pedigree, it means that individual's male. If you have a circle, that means the individual's female. Um, when you have two individuals that mate, there's a line between, so between the male and the female in this case. I've never seen it come up, but it could come up. Mating between two close relatives would be male and female with two lines between it. I'm going to jump over here for a second. I'm going to particularly focus in on this. A filled in square or circle means the individual is affected. Often we use pedigree charts to track diseases, genetic diseases, and who might be affected or not by that genetic disease. And so that's why we say affected. We might also say that filled in is dominant and not filled in is recessive, or the dominant phenotype and the recessive phenotype. Okay. I also note individuals whose boxes or circles are half shaded are also often referred to as autosomal heterozygous. In other words, meaning that they have both the dominant and recessive allele. That one doesn't turn up as often, I find as well. And so this is just a really simple representation of a pedigree, and I'll run you through how it works. So these two individuals, this female and this male, mated and had these three offspring. One of these offspring was male, one of these offspring was female, and one of these offspring was male. So they had two male offspring, one female offspring. None of these individuals were affected by whatever we might be looking at. Roman numerals symbolize generations. So this is generation 1, generation 2. Arabic numbers, that's these three here, symbolize the individuals in a given generation by birth order, meaning that this individual was born first, so this male was born first, this female was born second, this female or male was born third in that generation. Okay, so that's the general breakdown of how a pedigree works. To work with a pedigree chart, it's important to start to establish what you know about certain individuals. So here's how I play the logic in this. I often find the individuals that I can be sure of, and then I work my way back up if I can, or back down. So, for example, if we're studying a trait or a genetic disorder where only individuals who are homozygous recessive are affected, then I would go in and find all of the shaded individuals on there and fill in their genotypes. So little r, little r, little r, little r, little r, little r, etc., etc. And then I would start to figure out what their parents might have had in order to do that. So fill in suspected genotypes, see if your logic makes sense. If it doesn't, start again. Sometimes you need to do a Punnett square to, if you can't work it out in your head, to see if it makes sense. But if you've done enough practice with Punnett squares, you can almost start to do them in your head for these simple things. So here's an example that came out of your textbook. And we'll sort of work our way through this. together, and I'll sort of explain my logic if I can. So it says people with albinism. Albinism means they don't have normal pigment levels. They tend to appear lighter than normal. It's a recessive trait. Okay, so that tells me that individuals with albinism, albinism is an A, so I'm going to think of little a, are people that have albinism. Okay, so we're going to use the pedigree chart to answer the following questions. Use an uppercase A to represent the dominant allele, and a lowercase a to represent the recessive allele. So, first few questions. Let's play around with this. How many children do parents A and B have? Well, here's parents A and B. They're labeled on here. They mated, and then where these branch off, those indicate the children. So they had 1, 2, 3, 4 children. So we can answer that as 4. That is the most wacky 4 I've ever drawn. Now, the tricky part here is indicating the genotypes of the parents. And so this is where you want to try to reverse engineer. We're trying to figure out these genotypes. Here's a couple of ways you can tackle this. Okay. The first way is I would go through and I would say that we can figure out the genotypes of all of these ones that have albinism. So that individual is little a, little a. So is this individual. So is this individual. Okay, so is this individual, and this one too. Okay, so we already know those genotypes because to be an albino, to have albinism, you need to have both recessive alleles. So looking back at these parents though, okay, if you look, these parents were able to produce normal or non-albinic offspring, as well as offspring with albinoism. What that must mean then, and those parents are not albino themselves. So if they're not albino, they must have the dominant allele. But in order to produce offspring, they must also each have the recessive allele. And if that doesn't make sense, do a quick Punnett square to remind yourself how that could be. Right? Is they're going to produce individuals that are non-albinos, which would be these 3 individuals, but they're also going to produce an individual, or the possibility, a 1 in 4 chance of producing individuals who are albinos. If you do any other combination of parents, it's not going to work. If this parent, for example, was big A, there would be 0 chance of producing an offspring who has albinism. Okay, so that must be the genotypes of the parents. And what are the genotypes of M and N? Well, we've kind of half done our work already. We know that this parent here, this male here, and that female both have albinism, which means that the only gametes they can put forward in a cross are the little a gametes. So that means that N and M must both be little a, little a. There's no other way they could do it. If either of these had a dominant allele, they would be normal. They would not have albinism. Okay, so you can see how you can play with the logic in these pedigrees to try to get it to make a little bit of sense for you. So here's another example. It says label the genotype of each of the individuals below its symbol on the pedigree chart, and note that color is not a sex-linked trait. We haven't talked about sex-linked traits yet, so there's nothing to worry about there. Sex-linked just means that it's the gene is on the X or the Y chromosome, usually the X chromosome. We'd call it X-linked at that point. It tells us here brown eyes are dominant over blue eyes. So what this tells me then is that the shaded in individuals have blue eyes. You can choose what letters you want to use. Let's go, if it's eye color, let's go with E for the brown and little e for the blue. Okay, so then shaded in individuals have blue eyes. What that means is all of these individuals must be little e, little e. So that individual there is little e, little e. So is that one. Sounds like I'm saying Italy, Italy. This individual here that individual there, that individual there. They're all little e, okay? What it also means over here is that this individual here, all of these ones that are not shaded must have at least one dominant allele because they're expressing that. Now, we can't be 100% sure every time what the genotypes are going to be for all of these individuals. And this cross is interesting up here. These parents only had 4 offspring. All of their offspring turned out to have brown eyes. However, we can't be 100% sure that this individual is big E, big E. It could also be big E, little e. Because our sample size isn't large enough, there's not enough individuals here for us to come up with a really good idea of what these offspring are. But here's so we can't be 100% sure of that with this group. We know for certain that this individual here is big E little e, not big E big E, okay? Because they have the little e from this parent and a big E from this parent. We can also be sure here, okay, that this individual is big E little e. And why we can be sure of that is they're going to have a little e from this parent and a big E from that parent. The other way we can be sure is that these individuals are able to produce blue-eyed offspring as well. What we can't be sure of are the exact genotypes of these two. We know they have to be big E, but they could be big E, because that's a possibility for these individuals, or they could be big E, little e. So often I'll represent that as a big E and then a question mark. We can't be 100% sure. Same thing here. We can't be 100% sure what this individual is going to be, and we can't be 100% sure what these individuals are going to be based on actually, no, we can be 100% sure of these individuals because the mother is little e, little e. This one we're not 100% sure on, but chances are that individual, sorry, I lied. We can know what this individual is. I'm getting tired, can you tell? Because they have to provide, this little e has to be provided to that individual. This little e provided to that individual. Okay, so it's a logic game working with these pedigrees, and often you have to kind of work your way through your thinking a little bit before it all makes 100% sense. Okay, so that is the end of the first video for concept C5. This is the pedigree bit. If you wanted to go and jump into the questions out of your textbook, you can. I'm going to just jump back to this for a moment and remind you that you can also be doing, if you haven't done it already, this worksheet, which is dealing with the Punnett squares and giving you practice. You should have done it already when I asked you to pause. If you didn't and you just watched the video through, then by all means jump into it and do it now and do the little bit of practice with pedigrees out of your textbook. That is it for me for the first video for concept C5. You'll have the second video coming up subsequent to this that will cover the last bit, monohybrids. We're going to go into a little more complicated stuff after that. So as always, stay healthy, make sure you're taking care of each other, and we'll talk again very soon in the second video of this concept.